Christiane Nüsslein-Volhard. I went to the EMBL as one of the first round of group leaders in the new building at the 1st of February 1978.
And EMBL in those days, I imagine, was very different to what you see today.
It was just starting. I mean, it had been founded 4 years earlier, and I think the first round of group leaders already had labs at the DKFZ or other places in Heidelberg downtown. I joined a lab with Eric Wieschaus. We were Drosophilists. We were the two people who knew something about Drosophila, and another group with Vincent Perrotta. He wanted to start working with Drosophila, and this was the reason why we were hired.
I see.
Essentially to support Vincent. Yeah. But we ourselves didn't do any molecular biology, so we were sort of outsiders. We did genetics with these small beasts, and people didn't know anything about that, and were not really interested in anything. So we had a sort of slightly outside-ish role.
And did you already work with Eric Wieschaus before you came to EMBL?
I knew Eric, yes. I started working with flies 3 years earlier.
Okay.
In Walter Gehring's lab in Basel, where he had done his PhD, and we met, we overlapped briefly, and we had very many common interests. And actually, we had already early on discussed whether we should go and try to find a lab together, because we wanted to collaborate, and we were interested in early embryogenesis and genetics of embryogenesis. Strongly interested, and so we were actually. It was a great luck that we could join a lab there, and that we both were offered a position. We were not really group leaders because we had no group. We had two heads and had one technician. This was sort of, I mean, we were basically, I think, independent postdocs. Yeah. But we were thrown together in one small lab, which was so small that we couldn't even have our own desk for as an office. Yeah. It was a very small cubicle, and finally I got a room downstairs, and it worked out and so on. Somehow we squeezed in, and on the one hand it was a unique opportunity for both of us to have a position which was independent, and we could follow up our own research, because at that time this was really rare. Yes. Unlike in the States, where you had assistant professorships which you could apply for, in Europe usually people were research assistants to professors or directors or so. And they were weisungsgebunden in German, meaning they had to do what the director was telling them. Yeah. So they were not independent. Yes. Only in the Max Planck Society in Tübingen, where I had done my PhD, there was one institute with 4 junior group leader positions. And this was a singular and unique small institute. And the EMBL was also of the same shape, and this was pretty new, and great. Essentially a wonderful idea. And then I did a postdoc in Walter Gehring's lab, and I had freedom. I mean, I could do what I chose my project and did it, and then I continued with a fellowship in another professor's lab to do a second postdoc, and this guy also said, I don't care what you're doing because you have your own money. So having an own fellowship is also given you a certain independence. Yes, so I had first I had an EMBO fellowship, and then I had a DFG fellowship. So I was trying to keep as much independence as ever possible. Yeah. Because I hated being told things. Yeah. I mean, already at school. So I insisted on being independent. Yeah.
That's great, and it's actually a really good lesson, I think, and important to communicate to young scientists today, that rapidly building a lot of people that you're responsible for doesn't necessarily correlate with success, often inversely. When you're young and productive and able to work, being able to work with a small amount of administrative responsibility, but a lot of independence, is one of the best things that can happen to you.
Yeah, and then also we didn't have to care about money, but we were not very expensive, and we had a very small lab, so the status was really low. I mean, we were the lowest in status of all the group leaders, I'm sure. But we didn't care much. I mean, at the beginning, it sort of, I found it slightly irritating. Felt. Yeah.
But still, what came out of it was transformational.
Yeah, no, and also exactly. I mean, it was a piece of luck that we were put together in this small lab and shared a technician, so we had to agree on what we both did. We were not in the sense, we were dependent on each other.
Yeah.
And it was imperative that we kept peace, and then finally we realized it would be best to have a shared project.
Yes.
And then we did this big project, which none of us could have done alone.
Yes.
But together we were really, really strong. We were very strong because we had very sort of similar qualities with respect to observing and seeing. I mean, because it was it was a screen for visual.
Yes.
Perhaps
you could explain to us just a little bit about this transformational developmental screen you did with Drosophila.
At that time, it was not very. I mean, people worked on genetics in Drosophila because the adult fly had these fantastic structures which could be modified by mutations. So it was the organism to discover genes, because it's built with a cuticle, and the cuticle is decorated with tons of bristles and hairs and eye facets and legs and wings and so on. The embryo was not really much looked at because it was difficult to study. It was very tiny compared to other insect embryos, and people really didn't know what was in there and how it would determine early development. People had at that time this theory of the homunculus, perhaps you have heard of it. I mean that there is a pre-shaped organism in this egg which just has to develop, and in the egg already is a sort of a mosaic of determinants localized maternally, determined under the control of the maternal genome. And this was not proven, and it was a bizarre hypothesis, and people had tried to find mutants which would then alter from the mother already the future generations' wings and legs and so on. And they failed to find such mutants. On the other hand, other people had developed concepts of gradients, meaning and theoretically it was not possible that everything would be predetermined in the egg, because then the next generation also would have to be predetermined in this and the next and the next and the next, and this is not possible. So it means theoretically you have to demand that in each generation you have an increase in complexity. You start with a almost uniform egg, and then you build up a complex organism. But still, the idea at the time, it was maternal. And what we found out, essentially, that this was wrong, that the mother only put some gradients into the egg, not more than just anterior posterior and dorsoventral. And I had done research in Walter's lab showing that there was an anterior posterior gradient and also that there was a dorsoventral gradient. And for more information, we didn't have any evidence. And we found, Eric and I then got interested in mutants which were dependent on the embryonic's own genome after fertilization, and only 2 such mutants were described. Although people knew that there must have been many lethal genes, there were many lethal genes, and they also were lethal at embryos, but people thought that these many genes wouldn't be helpful to analyze, because probably the phenotypes would be messier, uninterpretable, or the embryos would just stop developing. So no one looked for these mutants. It's also difficult to do the screens, actually. Yes.
These are homeotic mutants?
Homeotic are the ones which affect the adult fly. I see. But the embryonic mutants, there were only one was known and described, which was transforming the cuticle, the ventral epidermis into nervous system, called Notch. And it had a very spectacular phenotype, had been early described already in 1950. And then I found in a textbook another one which made fewer segments. And this was very exciting because we didn't know the existence of such mutants, which had a very specific, in this case, a very specific phenotype. So not this, what people anticipated, messing up everything, uninterpretable and so on. And then we figured I mean, we have already 2 such mutants. Maybe there are many more.
Yes.
And maybe it would be really interesting to see whether we can find more. And actually then we designed, we first looked around and found some more just from the shelf, from some stocks or so. And then we calculated that we could do a saturation screen for the entire genome to get all the mutants with a specific phenotype.
And how did you metagenize?
We metagenized by feeding the males with EMS. This is acetylmethane an alkylating agent.
Yeah.
And then they would fertilize with mutagenized sperm, and then in the next generation you had heterozygotes, and from each heterozygote you had to establish an inbred family and then screen the inbred, the next generation, whether they produce homozygous mutant embryos. Yeah. And then we screened those mutant embryos by vision. I mean, they would not hatch. They would stop development because they were mutated. They showed mutant phenotypes. And we screened many such inbred.
And this wasn't screened by robots and AI, it was screened by...
No, I mean, you have to grow the flies, you have to cross the flies, you have to propagate them through several generations, and you do that in steps, of course, scattered over weeks. And the first screen lasted 3 months, but 3 months is not a very long time. Yeah, and we were 4 people. I mean, we could manage. It was very much work, but it was possible, doable. Yes, we were well prepared. We had prepared everything very well. Everything went smoothly. We had done a pre-screen. And we isolated actually over in a total of, I think, 1 or 1.5 years, isolated a total of 6,000 mutants or so, 600 mutants, and they defined a total of 120 genes.
Did you have
we have alleles? I mean, we had several, all the important genes were hit more than
multiple
once.
Alleles.
Mostly more than once,
yeah. And did you have a preconception going into the project of what type of phenotypes you were looking for? Extra bristles or more segments, or
well, we had this
was
one
there a big
mutant
surprise?
We had this one mutant with fewer segments. For exactly, I mean, it had a very constant phenotype, and then we had this other mutant with this lacking of the ventral cuticle. We knew the cuticle structure very well in the meantime. I mean, the larvae, I mean, what comes out of the egg is not the fly, it's the larva.
Yes.
And the larva has a simple structure, but it's also decorated with rows of segments, with a dorsal and ventral pattern, with some sense organs at the head and some sense organs at the tail. And so we could screen for subtle, also dramatic aberrations from the normal pattern. And we found a total of 120 genes where the phenotypes were clear aberrations from this normal pattern. Most of the embryonic mutants actually didn't have a pattern defect. They were just probably housekeeping functions where the embryos couldn't finish, or they couldn't hatch, they couldn't move. But there was a limited number, and this was also important because it would have, I mean, had it been 1,000 genes, it would have been very difficult to handle. So it was just 120. And then it turned out...
It was in the Goldilocks zone. It was in the Goldilocks zone, not too few and not too many.
Yeah. And we focused on the ones which were easiest to classify because they affected segment number and polarity. And this was easy to see, even for blind people. And you always say, you can count segments and you can see, and we had some. And we had a strange rule that every other segment was hit, and in one class of mutants in different manners, and then we had another class where the segments showed duplicated polarity of the denticle bands. Interesting phenotypes, pretty phenotypes. So they were not easy to interpret at that point, but one could see there is a system, and these genes are all important to make a normal larva. Yeah. So if you want to understand development of a fly embryo, you must know the function of all of these genes. Yeah. And this was done in these 3 years. We shared this lab at the EMBL, and we published one big paper on the segmentation mutants in Nature. Yes. 1980. Yeah. And then we separated. Eric went to Princeton and I went to Tübingen. and in our own labs finished up the description.
It must have been a very, I mean, it was obviously a very fertile project. It must have been a very exciting time as you discovered these different mutants. Do you remember any particular eureka moments of finding a special, unexpected one?
No, it was it was very exciting. I mean, the early excitement was when we found this rule. We called it 2468 rule because every other segment was skipped, which was completely unexpected and novel. And we thought, gee, I mean, we have to solve this puzzle, how to make segments. And we figured maybe it's a wave and you have a different wavelength or something, but it was very bizarre. We couldn't really figure out. And there was one, there was one Heureka. One day we found, I had worked on another system, dorsal.
Yes.
This was dorsal ventral polarity, and we had a mutant, and this was my first mutant I isolated already in Basel, which abolished the entire polarity and had only dorsal structures. Actually, yesterday, Patrick Kramer said he had made the structure of this protein. This was an NF-kappa-B homolog.
Yes.
Yeah. No, and this, but so. I knew these dorsalized embryos. They were just an empty bag of cuticle with lots of tiny hairs, and all this segmental pattern was lost. And then we found one evening a gene which had denticle bands circling around, only ventral structures, no dorsal structures. And I shouted toll. And this was the toll gene, which turned out to be also a maternal gene, also messing up the dorsal-ventral polarity in the opposite direction, a key gene for dorsal-ventral polarity. And it turned out later to be the key gene for innate immune system.
Yes.
And this was very famous. And it's the toll-like receptors, which became their name because we are sitting at night at this microscope. Look at this toll.
And toll, of course, is a German word for amazing.
Amazing.
Yeah. I mean, I have to say, I knew a great deal about your work, although I didn't work on Drosophila because as a graduate student in Cambridge, I was surrounded by people like Peter Lawrence, Gary Struhl, Bob Holmgren, and many others who all worked on these fields, and all they would talk about is your work and the importance of it.
Yeah, there was a very strong Drosophila Cambridge group.
Yes.
Yeah.
Yeah, so like gradients and developmental mutation. So I think it was understood extremely quickly by the field how transformational this screen you'd carried out was.
No, not very quickly. At the beginning, when we had this zoo of mutants, people were just stunned.
Yeah.
They said, what does it mean? It was a change of paradigm to go from the maternal pre-homunculus idea to
yes,
it's the embryo who does the thing. And the embryo must have lots of mechanisms which requires these genes to build these structures. And but at the beginning, people were just confused, and they appreciated it and said, "Oh God, this is very interesting and very exciting," but they couldn't make much out of it. Yeah. I mean, we had the reviews for the Nature paper said this is the most exciting paper for years in developmental biology.
Yeah.
And it was clear that maybe using mutants you could eventually understand how segments are made because people had no clue.
Yes.
No clue.
Yes.
Whatever.
Well, it's clear your work did provide the building blocks for a large part of the field of
I mean, the
biology.
breakthrough was essentially when people had developed methods to clone genes in Drosophila.
Yes.
And the first genes were cloned were the homeotic genes, and these were the ones, before we did the screen, these were the only ones which were known as developmentally important genes. So the bithorax complex and the antennapedia complex, and they were cloned by Hognes in Stanford and by Walter Gehring in Basel. And they discovered the homeobox, which was a big excitement, showing that there was this homology between vertebrates and invertebrates because they were present, they all had this homeobox. And this was very exciting, although in retrospect one can't really, why did people believe that the development was so different? Because they must have come from a common ancestor.
Yes.
Bilaterian common ancestor.
Yeah.
And that it dawned on people, maybe already this bilaterian ancestor must have had these genes. Yes. And then when the first genes were cloned, one could find homologs in humans, in mice, in invertebrates, and this made the excitement. So we found all the signaling components, the receptors and the ligands for the signaling components, which have been cloned in many labs then. And we founded a whole Drosophila community which worked on our genes. Yes. 120 is a lot. Yeah. And they are now cloned. They are all cloned now. Yeah.
No, it was a phenomenal achievement and tremendous. And then, of course, you've gone on to great things, and you've got the satisfaction of seeing that you helped to really drive the field of developmental biology. But I think it is a great lesson as well, as I say, for young scientists starting out today, that you don't have to start off with a gigantic lab and huge amounts of money, and you can still do very important things if you work on important problems.
Yes, and we were Drosophila at that time it's a wonderful organism, and it turned out to be so helpful. Yes. And there is no better organism. And people actually, at that time, there were still people who thought maybe we should use another fly, or we should use another organism. And Caenorhabditis had been invented by
Sidney Brenner.
By Sidney Brenner. He designed it from scratch, essentially. But Drosophila turned out to not only have a wonderful genetics, only few chromosomes. Yes. Had giant chromosomes where you could visualize the position of the screen of the genes, which was incredibly important for the cloning purposes. Had insertion, had these jumping elements where you could make insertional mutations to clone the genes, which is also wonderful, And then the embryos. This was my concern when I had done my PhD on RNA polymerase in bacteria, essentially, and then have chosen to do developmental biology because I found it fascinating how you can get out of very unstructured something, and I wanted to combine development with genetics because I had understood from work of other people who'd done saturation screens on replication and other things, that the mutations in genes were really crucial to define a gene. And in contrast to cell biology, where you grind up things and so on. So this grinding on transplantation experiments in embryos were very unsuccessful. They couldn't find the factors. It was just too much artifact and so on. And I figured that maybe if you have a mutation and you knock out the component, the morphogen or whatever, with the mutation, and then you add extract from the wild type, this would be the ideal assay. Yeah. And this was what I essentially at the beginning wanted to do. Yeah. And which means I had to find mutants. Yes. And then I wanted to find maternal mutants for the gradients. Yes. And did screens for maternal mutants. Yeah. And then when we, together with Eric, we saw that the zygotic genes were easier to get. We switched to the zygotic genes first. But then when we had our own labs, he in Princeton and I in Tübingen, we continued doing the screens for the maternals. Yes. So I did then big maternal screen on the third, and Eric and Trudy, his wife, did a screen on the second, and finally we ended up with 30 genes for the maternal, and they were all defining gradients, essentially. Yes. Yeah.
Yeah, and of course the work culminated in you receiving the Nobel Prize, which was a wonderful recognition of its fundamental importance. Janne, as you know, we are now in the 50th anniversary year of EMBL as an organization, and we're here celebrating that fact, and it's been great hearing about the wonderful science that is still going on at EMBL, and hearing from people such as yourself about the wonderful science that was done in the past. Perhaps you could say a few words about your own memories of EMBL, of what EMBL meant to you in starting your career, and possibly share some words of advice for young research scientists starting out today.
This advice for young research scientists. People ask you all the time, I don't know. I think you just have to love what you're doing. And you have to be really, really very much driven by finding out. I'm very impatient, for example, and I rushed to the lab and in order to, when there was a cross the next morning, the embryos were ready to look at it. It's very exciting, and the rich surrounding here at the EMBL, I think this is really a great attraction.